Thank you, Mitch. That's a really kind introduction. Eh bien, merci Geneviève pour l'invitation. Merci beaucoup. Thank you for to the organization committee for this invitation, and I'm delighted to share our work today on, as Mitch mentioned, bioactive metabolites involved in sex-specific differences in the immune system and immune-related diseases. And today we'll talk a little bit about a couple, but at least end up with Alzheimer's. So a bit of an inflammation skew, if you will. So preaching to the choir, admittedly, the challenge is that diseases affect men and women differently. And this, as we saw this morning and throughout the afternoon, there are profound sex-specific differences in the biology of men and women, including in the immune system. And many immune diseases and conditions affect men and women differently, not only in terms of their incidence or prevalence, but also their pathophysiology and severity. However, most treatments, if they exist, since we're talking about autoimmunity today, are one size fits all. And understanding, we think here in my lab and at LJI, that understanding sex-specific differences at the molecular level, as we've discussed today, can uncover new disease origins, lead to better prevention mechanisms, and hopefully deliver on the promise of personalized medicine that Geneviève mentioned this morning. Preaching to the choir again, at least it's a different format of the graph, okay? So I can save some time and just say that yes, we've established today that at least certain autoimmune conditions such as Hashimoto's, lupus, and Sjögren's syndrome, are more prevalent and their incidence is higher in women compared to men. And even when we go down to the autoimmune diseases that are more common, like rheumatoid arthritis and multiple sclerosis, there's still that skew. And if we can ignore this, at least let's talk about what's the consequence of that. And that is that autoimmunity today in the US and globally is a leading cause of morbidity and mortality in girls and women. And perhaps terrifyingly, cases are increasing mostly for most of these diseases. And some of them, yes, are rare diseases, but collectively as a whole, they certainly are not. The sex dimorphism that is observed in the biology is caused or must be influenced by as yet unknown differences in the immune system. And as has been a theme today, we think that this is a new frontier in immunology research. So again, in general, we can say that women are prone to autoimmune and inflammatory diseases, and in general, men lack a comparatively robust response to pathogen. And again, preaching to the choir, because this was so well introduced in the first talk, as well as subsequent talks, we've known that some of this relates to differences in immune cell numbers in some of these important innate and adaptive immune cell populations that we've discussed today. COVID is kind of an interesting example because it very well illustrates the generalization that I just presented. Acute COVID displays globally, and we saw that this morning as well, 10 to 20% higher mortality rate for men globally. This is data from our own local cohort. It's called the SoCal Coral study. This is a collaborative project that we collaborate with Dr. Susan Cheng at Cedars-Sinai Medical Center. And we've studied a relatively small cohort of COVID-19 patients during the first wave. And what we found was that, tracking with what others have reported globally, indeed, that if we look at the hazard ratio for COVID-19 disease severity, taking into account the key covariates of age, BMI, and sex, that male sex in both the ICU patients as well as the intubated patients was the strongest predictor of severe disease, or had the strongest association. And this was corrected across the board for comorbidities. So just trying to convince you there. And with respect to the immunology of that, and we talked about the type 1 interferon antiviral response in the last talk, what we can see here is that with respect to these essential antiviral interferon-stimulated genes, and this is looking at RNA-seq data from COVID-19 infected lungs, looking at the general expression of several interferon-stimulated genes, that in the males, compared to the females, these are also lower. And this might be related to key innate immune genes such as TLR7, which are expressed on the X chromosome, or also indeed to other innate immune disparities between the sexes. In contrast to what we talked about acute COVID, the data is emerging and suggesting that women are more likely to experience the symptoms of long COVID, which are more similar to an autoinflammatory, perhaps a real autoimmune disease, certainly with profound neurocognitive involvement. And the molecular drivers of this, not well understood. So in my lab, as Mitch mentioned, we profile small molecule metabolites in the blood and tissues in order to understand the molecular basis of inflammation. And the reason that we focus on metabolites is because lying at the very end of the central dogma, as they are, we think that they are intimately linked to disease processes. Not only that, but Geneviève talked about diet and environment being an influence on our immunology. And in fact, our small molecule metabolome in our blood and even in our tissues is a readout of our cellular and tissue physiology, our diet, and also our gut microbiome. So again, we think that by studying the metabolome or metabolites in the blood, we can uncover closely the molecular underpinnings of disease. This isn't always a very easy endeavor because the dark side or the truth is that the metabolome is not really well defined. There are thousands of small molecule metabolites in our blood, and we really only know the chemical and functional identity of a minority of them, maybe 10%. So practically, as a scientist working in this space, it can often be frustrating for myself or folks in my lab to be studying molecules that we know are important through the biostatistics in a particular disease, but we might not even know what they are, much less what they do. But that said, we do rely on reliable, rapid, microscaled quantification of metabolites in blood as well as in other biosamples, tissue homogenate or cell supernatants. And using a automated platform, we can, hooked up to a mass spec, we can analyze quite complex samples. Here, this pseudo representation of blood, first separation of many small molecule metabolites in the sample, and then they're analyzed in the mass spec. And then in real time, we can detect each separated molecule as a distinct peak on the mass spec. Now, this is a plasma sample, human plasma, and you can see that there are many peaks crunched all together, and then we rely on software and AI to go in for reliable peak identification and relative quantification in the sample. So this is just another representation of that. Each separate colored peak represents a different small molecule metabolite in our blood, and as you can see, there are overlapping peaks, and we require the software to separate each one individually and calculate the area under the curve, which is proportional to its relative concentration in blood. I hope I can convince you here that there's quite a dynamic range of detection for each molecule of interest. And in terms of what types of molecules we can detect, quite a range, from polar metabolites to non-polar metabolites and lipids. We personally like to study bioactive lipids because they're signaling lipids, and because of that we know that they have a role not necessarily only in bioenergetics but also in signaling and in the immune system. And we can detect these quite reliably with a relatively low coefficient of variation. So Mitch mentioned, how we got into this space and thinking about metabolites and inflammation really stemmed from a study that we performed, a collaborative study, where we identified and characterized an anti-inflammatory metabolite, or lipid, in the blood of patients, cancer patients, receiving cancer immunotherapy. And what we found was that in patients who had relatively high levels of this lipid metabolite, it's a fat actually, and it can be supplemented in the diet. It's called LPC. And patients with high LPC were observed to have no or little toxicity when they receive immunotherapy. Now remember that immunotherapy boosts the immune system, but it can also cause unintended inflammation or autoimmunity. And so high metabolite LPC, no inflammation. In contrast, we found that low levels of LPC were observed in patients who developed immune-related adverse events, inflammation or autoimmunity, after their immunotherapy. How did we set up the study? We obtained patient plasma from 3 different cancer patient cohorts, as well as plasma or blood from healthy cohorts, and we analyzed them, similar to what I showed you 2 slides back. And then we were able, once we had calculated the area under the peak for each small molecule metabolite, we integrated this data statistically with the patient outcomes data. Very simple, no inflammation or inflammation. And we were looking for things, metabolites, that are high in the non-inflamed or vice versa. And this way, we were able to identify candidate autoimmunity metabolite correlations. And this is where the functional studies came in. I'm just showing you a little bit of the data that came out from one of the cohorts. These are melanoma patients who received anti-CTLA-4 immunotherapy. And what we found was that there was really one metabolite that was strongly associated with the development of autoimmune or inflammatory complications after immunotherapy. It's actually, you can see that it's on the negative axis, and what that means is that every single patient who developed an autoimmunity had lower levels of this metabolite, making us think perhaps therapeutically, if the autoimmune patients are missing this metabolite, maybe can we put it back in and see an effect in suppressing inflammation. And indeed, to do that, we turned to mouse models of colitis, and we used 2 different mouse models of colitis. One in a mouse knocked in for human CTLA-4, which can be treated with ipilimumab, which is the human anti-CTLA-4 antibody, and it develops multi-organ inflammation, but colitis, and then also a DSS chemically induced colitis model. And we did this with Mitch Cronenberg's lab in collaboration. So we put the lipid back at the same time that we induced colitis in the mice. And just looking at the histology score here, what you can see is that when we supplemented with the lipid, that the inflammation, the toxicity score, was suppressed, indicating that yes, indeed, inflammatory lipid metabolite was protective in colitis in the mouse model. To get a little bit to the mechanism, we also found that in these same mice that were treated with the anti-inflammatory lipid, that there were relatively lower levels of a pro-inflammatory cell in the blood called a neutrophil. So lower levels of pro-inflammatory neutrophils were also observed after administration of the lipid. And we do believe that the bioactive lipid is likely directly regulating in a cell-intrinsic manner neutrophil survival and pro-inflammatory functions. So, really with this study, this was our first foray into this idea that we can profile blood metabolites and relate them not only to clinical outcomes of inflammation, but also to fundamental immunology. So I'm going to switch tracks here and really focus on where we are now. And that is really looking at neuroinflammation and neuroinflammatory disease. And this interest was brought to me in a large part by a very talented postdoc in my lab, Priyanka Saminathan, who's a neurobiologist and has a neuroinflammatory background. And Pri really wants to understand, what underlies the observed sex differences in neuroinflammatory disease. For example, Alzheimer's and frontotemporal dementia are skewed towards biological women. In contrast, Parkinson's and ALS are skewed towards men. And of course, this relates to neuroinflammation, but again, the molecules and the cell types involved, not really known. To talk a little bit about Alzheimer's disease, women are really at the center of this disease problem. In that 1 in 6 women versus 1 in 11 men are at risk for developing dementia or AD during their lifetime. And that's even when we correct for increased age or increased lifespan in women. Not only are women more likely to develop Alzheimer's disease during their lifetime, but they're more likely to experience more severe disease, in that the degree of plaque formation and neurofibrillary tangles are more pronounced in postmortem. And this is accompanied by greater brain shrinkage and more rapid decline of memory and cognitive function. And again, we know that the factors include aging, oxidative stress, and the sex hormones, but really the precise molecules that are involved are not known. So we set up a study that was very similar to our cancer study, just swapping out the cancer cohorts for longitudinal cardiovascular or neurovascular cohorts. And we have a cohort called ARIC, which has enrolled participants from Eastern and Central USA. You can already see in ARIC there's a skew for dementia. The female skew in the dementia prevalence is already there. We've also collaborated with the FinRisk cohort, which is a longitudinal study of community dwellers in Finland. And then we're also recruiting samples from the University of Washington Alzheimer's cohort. Only right now a subset of ARIC has undergone metabolomics profiling, but we have been able to perform some preliminary biostatistics in the male and female groups with separation in order to identify sex-specific AD metabolites. And before I show you the data, I just want to talk a little bit about the clinical translation aspect and how we calculate this, how we look at this, how we measure this. We calculate a biostatistical measure called a hazard ratio. And we're looking at the relationship between the level of a particular metabolite either in the diseased population or in the disease-free population. And that comes out as what's called a hazard ratio. Now in black, you have a hazard ratio of 1, and that's neutral. That means that there's no association between the level of that metabolite in either the diseased or the undiseased population. If the hazard ratio is above 1, what does that mean? That means that that metabolite was specifically high in the diseased population, but not in the disease-free population. And that's significant. And what does that mean? We call that a pathogenic molecule because it's high in the disease subset. And what does that mean practically? That means that this molecule could be a biomarker, for example, if clinically translated. In contrast, if the hazard ratio is below 1.0, that means that this particular metabolite is low in the diseased population or is low in the disease-free population, but high in the diseased population. It's high in the disease-free, I should not be doing this late. A protective metabolite is high in the disease-free population. And what does that mean? Like LPC, this could be a potential supplement. So here's the data from ARIC. We looked at first, what I'm showing you now are the male-specific metabolites shown in blue. And we were really surprised. They're all pathogenic, at least in this subset of ARIC. We were disappointed by this because we expected to pick up protective metabolites, but we did not. All of the male-specific metabolites detected in the ARIC cohort are pathogenic. But what are they? Well, we have been able to define a subset of them, and they correspond to what are called pro-inflammatory leukotrienes. Leukotrienes are lipid mediators that are produced by the cells in our blood it's already known that overproduction of leukotrienes is involved in inflammatory conditions like asthma, rhinitis, uveitis, and other inflammatory conditions. So that kind of gives us a clue that we're on the right track for these pathogenic, perhaps neuropathogenic or neuroinflammatory lipid mediators, because leukotrienes are already known to activate immune cells in the blood, including neutrophils and also other subsets. So then leads us to ask a question, are higher leukotriene levels in the blood a biomarker or maybe even a modulator of neuroinflammation in Alzheimer's? And Pri and Maya in my lab have looked at the effects of treating microglia, which are macrophage-like cells in the central nervous system, with the AD-associated leukotrienes. And indeed, what we see is that these are pro-inflammatory mediators that are causing pro-inflammatory cytokine expression in microglia cells. So we do think we're on the right track there. But what about the female-specific metabolites? And this was also kind of interesting, and those are shown in red. Surprisingly, we picked up pathogenic molecules, but we also picked up protective molecules. And this was a shock, because we didn't expect this. Looking at the higher incidence of AD in females, we thought, well, why do they have protective metabolites? But then we looked at the data in more detail, and we realized that these protective metabolites are higher in the blood of younger women and are lost in older women or are absent in men, leading us to think that younger women possess immunoprotective, maybe anti-inflammatory molecules that as we age likely go down and may expose them to inflammation, vascular inflammation or neurovascular inflammation. What are these protective metabolites? We've been able to discern that they correspond to omega-3 fatty acid metabolites. And again, this bioactive function of these metabolites tells us that we may be on the right road, because omega-3 fatty acids are essential fatty acids that are naturally found in the oils of fish, hemp, and blackcurrant. And we already know that they affect brain homeostasis and development. And these are, of course, they're dietary supplements for both human and pets. And this is a billion-dollar industry. However, I'd just like to stress that the specific protective metabolite in fish oil are not necessarily known right now. And we've picked up specific metabolites that we think might give us clues to what is the real bioactive component of fish oil, which itself is quite a complex fat. And the question here would be, therapeutically, thinking down the road, can these specific fatty acid supplements protect older women and men against developing dementia or Alzheimer's disease? And to do this, we really have to go to look in vivo. And Pri has been working on developing a robust in vivo model to study Alzheimer's disease in the mouse model. And she's working with the AD-prone mice, such as the 5X FAD model. Now, this is a very aggressive model that is modeled after 5 different genetic polymorphisms that are found in the human. And she's really built this from the ground up, in the sense that she is focusing on microglia and developing here at LJI, with participation of our wonderful imaging core, precise methods to image microglia. And you can show some of our early pictures here, where each green dot is a microglia, shown here. And you may not be able to see it here, but in the 5X FAD mice, not only, there aren't necessarily, which are AD prone, as they develop Alzheimer's disease and show cognitive defect, their microglia change. And you can see here that they increase in area, circumference, and also expression of certain molecules like CD68. And what we're heartened to see in this model is that the females, shown in red, actually have worse pathology compared to the male mice. So we think this is a good model to study some of the molecules that we've pulled out from Eric. And that's where I'll end, conclusion and what's next. I hope I've convinced you that metabolomics, even unbiased metabolomics, while sometimes depressing, because you don't know what the molecule is necessarily, can link inflammatory molecules to diseases like cancer and AD. That metabolite disease associations can be pathogenic, biomarkers, or protective, treatments or supplements. And that we're developing in vitro and in vivo techniques to study brain inflammation through activation of microglia, these poorly understood immune cells in the brain. And that validation of AD-associated metabolites is an important step in their therapeutic development as biomarkers or as supplements. And I'll just end there and just thank the folks. I think I thanked the folks who did the work, but also our lab, our donors, and our other funding sources. Thank you.